Hi, my name's Will Krozik, Scientific Services Manager at Chemical Computing Group. Today I'd like to introduce MOE Project. MOE Project is a collection of 3D structures that are annotated and superposed to a common frame of reference. This automates the complex process of organising and standardising structural data so we can share it with project teams to leverage in their structure-based drug design. MOE ships with 21 highly curated families, and we can create custom families for any given target of interest, whether that's a protein ligand complex, antibody, CYP, peptide, RNA, for example. In today's session, we're going to take a closer look at the protein kinase family, and you'll see here at the top of the panel a list of the 11,000 or so members of the family, together with options to filter and refine our search at the bottom. And if I just click on one of these to have a look, you'll see that we're provided a PDB code, information such as the species, resolution. We get a reference, and we've provided links here for further reading. And if I just scroll along, you'll see that we also have information, in this case for DFG and alpha C in and out. Now, next, I'd like to get a feel for the structures and how these are organized. And I'm going to do this by just browsing through the family. And I'll just click to auto cycle through so that you can see the common rendering that we've applied. So for example, you'll see this green ribbon here at the top of the pocket. And if I click on legend, we'll see that that green relates to the G loop. We've also provided annotation of other important motifs like the hinge and gatekeeper shown in gold. We get a 2D depiction of the ligand when it's available. However, some of our structures are apo structures. When we do have a ligand, we can keep track of the ligand properties by adding some descriptors here so that they're displayed in the browse panel as we browse through. Now, an alternate way to have a look at and get a feel for the entire data that we have in the family would be to look at the statistics. If I click on the drop down, you'll see by default it's showing us the species, but there are other parameters that we can understand the distribution of. But looking at species first, we see that human is by far the most prevalent. So what we could do is add some filters to our panel so that we can filter down looking for just the human structures, and perhaps we've also got a subfamily of interest. So I'll select that, hit search, and we see that our search is refined to show us the 25 structures that meet the search criteria. Now I can also add in expression language, so I can use MOE's expression language here, or perhaps use one of the presets to search for any covalently bound ligands, and then I could load them all in, but perhaps I'll just take this one here with the best resolution, and I'll just load that into the MOE window for further analysis. So now that we have an overview of the family, let's think about how to apply the database for different efforts. For example, we might want to find templates for homology modeling, think about selectivity analysis, or mutagenesis. I'm going to start with mutagenesis, and if I look in the MOE window, you'll see that this particular ligand has an interaction. It's got a H bond with this residue 320. It's a threonine. I happen to know that this is the gatekeeper of the hinge, and this might lead us to question, can we always expect to find a threonine in this position within the sequence? To answer that, I'm going to go back to the stats panel. I'm going to change to residue UID, and I'm just going to hit the target button, which is going to return information on the residue that we have selected in the MOE window, in this case, threonine. And when I do so, we'll see that it's actually the methionine that's more prevalent in this position. So perhaps we'd want to consider performing some virtual mutations on this site to see how that implicates binding. I'll just clear the selection that we have and go back to the entire database, and then think about some other useful workflows. We could think about similarity searching, and we can do this for either the receptor or the ligand. In this case, I'm going to start with a ligand. So I'm just going to select the ligand, and I'm going to search similar to what we have selected. There's a default Tanimoto cutoff of 60% that's applied, but I can change this, so perhaps I want to be a little more stringent, and I can go ahead and search to return those ligands and the targets that they bind to, allowing us to think about things like selectivity. MOE project databases automatically have PLIF, or Protein Ligand Interaction Fingerprints, calculated, and this is ideal to leverage the organized structural data for statistical analysis. PLIF helps us understand how ligands interact with the aligned family by identifying key interactions, the type of interaction, or we could use it to elucidate pharmacophoric features. The PLIF panel has a barcode type representation at the top, which is telling us, does this have an interaction, yes or no? The bottom part gives us detail on the type of interaction, and you'll get a list of the residues along the middle. So here I can quickly identify that residue 323 has quite a few contacts associated with it. And if I just scroll along to residue 323, I can see the first entry has an AA type interaction. So I'll just raise bubble help, and that will tell me that lowercase a means we have a backbone hydrogen bond acceptor. Now, we also have a selector, which can help us filter for specific interactions. So here what I can do is I can restrict our search to the pre-filtered results that we have, and then choose the type of interaction that I'm looking for. So I could search for any interaction on 323, or I could be specific and say show me only the backbone acceptor interactions. I do have Boolean operators, so I could make my query a little more specific, but for now I'm just going to look for those acceptors, and perhaps I'll toggle to see these ligands within the MOE window also. So while we're thinking about similarity searching, the other thing that we may want to do is query the receptor. So I'm just going to load that into MOE. I'm going to load in the sequence of the structure that we have in the main MOE window. I can query based on similarity or identity. And this is useful when we want to start to think about off-target interactions. Perhaps I want to find a template for homology modeling or investigate the flexibility of a loop. So here I can see that we found a number of identical structures together with a couple that are very similar. So perhaps what I'd want to do from here is load them into the MOE window for closer inspection. And with that, it brings us to the end of today's session. Here we reviewed MOE project families as an automated process for collecting 3D structures and their associated properties within a common frame of reference to share with project teams. The resulting family allows for easy comparison of homologous structures and assists SBDD workflows with tasks such as identifying key interactions, selectivity, or mutation analysis. If you have any questions, please do reach out to us at support@chemcomp.com.